Greetings everyone, and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled, Get Rid of the Noise, Targeted Depletion Approach for RNA-seq. I'm Jeff Bogliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. RNA sequencing, or as it's often called RNA-seq, is revolutionizing the field of transcriptomics. This immensely sensitive technique reflects the dynamic nature of gene expression and captures an immense array of biochemical complexity. Yet, for all its intricacies and power, the one thing that makes this next-generation sequencing technique so interesting is the very thing that makes it challenging, RNA. Researchers using RNA-seq are often faced with uninformative reads that are strewn with high abundance transcripts, contaminated with ribosomal RNAs and cellular biomolecules not relevant to their current study. Investigators are always on the hunt for interesting new methodologies to clean up RNA-seq libraries of unwanted transcripts, increasing the specificity and efficiency of their sequencing results. Let's meet our panelists for this GEN webinar, who will discuss some novel approaches for the targeted depletion of specific transcripts from RNA-seq libraries using insertion-dependent adapter cleavage technology. Our first presentation will be given by Ashish Saraya, Product Manager at NuGEN Technologies. Dr. Saraya will introduce us to NuGEN's novel technology that utilizes insert-dependent adapter cleavage technology to provide targeted depletion of unwanted transcripts. Our second presenter today will be Maureen Peterson, Research Scientist at NuGEN Technologies. Dr. Peterson will take the technology a step further and describe a new methodology for the targeted depletion of ribosomal RNA for single-cell RNA-seq analysis. Before our speakers get started, I want to encourage the audience to submit questions for a Q&A session at the end of the presentations. We'll try and answer as many questions as we can, so simply type your question in the Q&A box on the left-hand side of your screen and hit submit. So, without any further ado, let's begin our webinar and open up the floor to our first speaker. Ashish?
Thank you for that introduction, Jeffrey, and to Jen for organizing this webinar. RNA-seq is rapidly becoming a standard experimental approach in biological research to answer a number of basic questions. The RNA-seq workflow starts with RNA isolation from a variety of sources and sample types. The isolated RNA is converted to double-stranded cDNA and ligated to specific adapters to generate libraries ready for sequencing. RNA-seq, however, still presents some challenges. One challenge is the presence of highly abundant, uninformative reads, such as ribosomal RNA or other highly expressed transcripts, such as globin, actin, housekeeping genes, etc. The presence of these uninformative sequences increases the cost of sequencing and data analysis. Uninformative reads can also interfere with data analysis by reducing useful information and introducing bias. Upfront transcription depletion methods, such as poly A selection and hybridization capture, have been designed to remove these uninformative transcripts. These techniques, however, require higher inputs, have limited effectiveness against degraded inputs, can introduce bias due to off-target effects, and are difficult to customize. The insert-dependent adapter cleavage, or InDA-C mediated depletion of uninformative transcripts, occurs after adapter ligation. Therefore, manipulation of the input RNA is not necessary and the transcriptome profile is not perturbed. This workflow also reduces the amount of input necessary for library construction and is suitable for degraded inputs. Finally, the InDA-C method can be quickly and easily customized to target any transcript for depletion from any organism. In the InDA-C workflow, after adapter ligation, the double-stranded libraries are denatured and probes specific for the transcripts targeted for depletion are added. The probes anneal to their target transcripts and are extended through the insert into the adapter sequence. The requirement for the probes to hybridize and extend allows for greater specificity in targeting the transcripts for depletion compared to hybridization alone. After probe extension, The resulting double-stranded DNA is then cleaved by restriction enzymes, effectively removing this molecule from the library. This method has several benefits. Informative transcripts are enriched by eliminating unwanted and uninformative transcripts. The depletion occurs after library construction, so the original RNA population is not perturbed as with upfront depletion methods. Depletion is highly specific for the targeted transcripts. And finally, probes can be quickly and simply designed to target any transcript from any species and incorporate it into the existing workflow. This provides a single workflow that is compatible with sequencing studies from any organism. Targeted depletion of uninformative transcripts can be useful for a number of potential NGS applications. Ribosomal RNA is typically the most abundant transcript and is the main target for depletion. Aside from depletion of ribosomal RNA, however, removal of highly expressed transcripts, either from tissue-specific samples or from model organisms, increases the number of informative reads. This can result in increased coverage of critical genes and provide a greater insight into the underlying biology. Specific depletion of uninformative host or both host and pathogen transcripts in host pathogen studies can increase the coverage of the host and pathogen transcriptome, providing better understanding of the host-pathogen interaction, as well as increasing the sensitivity of pathogen detection. Depletion of highly expressed transcripts can increase the dynamic range of transcripts detected during whole transcriptome or mRNA-seq studies. This provides more information on low or rare transcripts at the same sequencing depths, facilitating the discovery of reference and novel genes. In a number of higher plants, an estimated 50 to 80% of the genome is composed of repetitive elements. Sequences from these regions are difficult to align to the genome and can affect data analysis. Depletion of these reads can provide better results as well as allowing higher multiplexing and lower sequencing costs. Finally, chromatin accessibility studies typically suffer from a high prevalence of mitochondrial DNA reads. Depletion of mitochondrial DNA reads can increase the number of informative reads, allowing for simpler data analysis and more precise conclusions. Blood is an attractive sample type for human disease studies due to the ease of collection and high yield of intact RNA. This sample type, however, presents unique challenges, specifically the high abundance of ribosomal RNA and hemoglobin mRNA, which can represent over 60% of a sample. We constructed whole transcriptome RNA-seq libraries from 4 blood samples without any transcript depletion. The sequencing results showed that ribosomal RNA represents between 37 to 67% of the total reads, depending on the individual. Globin mRNA represented between 13 to 31% of the total reads. This means that only 20 to 38% of the sequencing reads are informative and provide relevant information. Targeted depletion of these uninformative reads from ribosomal RNA and globin using InDA-C reduced the percentage of ribosomal RNA reads to between 5 and 14% of the total reads, and globin reads to less than 2% of the total reads. With the depletion of these transcripts, informative reads were increased by an average of 61% for these samples, providing more useful information and increased coverage. Tripling the number of informative reads can also allow for greater multiplexing during sequencing, effectively reducing sequencing costs. Visualization of the globin coverage, both without depletion, outlined in green, and with depletion, outlined in red, show a significant reduction in reads from all 4 targeted transcripts after InDA-C treatment. This depletion is consistent across the entire length of the transcript, indicating that there is no sequence-specific bias during the depletion. Additionally, correlations between InDA-C treated and untreated samples were consistently high for all 4 samples, indicating that the transcript depletion had minimal impact on non-targeted transcripts and preserved the sample biology. Many clinically relevant patient biopsies are formalin-fixed and paraffin-embedded to maintain morphology. Each FFPE block, however, represents a finite resource with a limited amount of material available for study, so methods requiring high inputs cannot easily access these samples. To compound the problem, FFPE preservation was not designed for nucleic acid preservation, and RNA and DNA isolated from these samples are typically degraded. To test the efficiency of our system with FFPE samples, we constructed RNA-seq libraries from 6 FFPE samples derived from human colon. The FFPE blocks ranged in age from between 8 to 12 years old, and the isolated RNA had RIN scores between 2.1 and 2.5. 100 nanograms of each FFPE sample, along with 10 nanograms of a high-quality RNA, were used as input for library construction. All samples showed good alignment, coverage, and strand retention, even with minimal inputs of degraded RNA. Importantly, even with highly degraded samples, we see significant ribosomal RNA depletion. Next, we looked at the effect of InDA-C on differential gene expression. The differential expression of over 400 genes were analyzed by TaqMan. A comparison of differential gene expression of these genes between InDA-C depleted RNA-seq and TaqMan showed excellent concordance, indicating that the InDA-C method did not perturb gene expression. The InDA-C-mediated transcript depletion can be quickly and easily customized to target uninformative transcripts from any organism. This can include specific model organisms that are used for studying basic biology or human disease, to whole microbiome studies of the human gut or deep-sea vents, as well as non-model organism studies used to study and identify novel biological pathways or processes. We constructed RNA-seq libraries from RNA isolated from 3 model organisms, mouse, rat, and Drosophila, both with and without InDA-C ribosomal RNA depletion. All libraries showed excellent strand retention. In the absence of ribosomal RNA depletion, the ribosomal RNA reads represented between 35 to 86% of the total reads for these samples. InDA-C-mediated depletion of the species-specific ribosomal RNA transcripts decreased the number of reads to less than 10% of the total reads. This represents an average ribosomal RNA depletion of over 90%. The elimination of these uninformative reads can increase the dynamic range of transcripts identified, result in the identification of additional novel or rare transcripts, allowing for greater insight into the biology. P. gingivalis is a gram-negative pathogenic bacterium found in the mouth and implicated in periodontal disease. In the absence of any depletion, ribosomal RNA represents approximately 90% of the total reads. Species-specific ribosomal RNA sequences were used to design custom InDA-C probes. Use of these custom InDA-C probes reduced the ribosomal RNA reads to 10% of the total reads. The increased number of informative reads can allow greater multiplexing, significantly reducing sequencing costs. This also highlights the customizability of the workflow, which can be tailored to any organism or sample type. NGS can provide a wealth of information on the transcript profile of a sample. Low expressed or rare transcripts, however, can be difficult to detect due to the abundance of highly expressed transcripts. Detection of these rare transcripts require very deep sequencing to overcome the abundance of reads derived from highly expressed transcripts, resulting in increased sequencing costs. While eliminating ribosomal RNA transcripts can help, depletion of other highly expressed transcripts can further increase coverage of rare transcripts, reducing the depth of sequencing needed for detection. We initially tested the ability to increase detection of rare transcripts using the ERCC spiking controls. ERCC controls are a set of predefined polyadenylated RNA transcripts with known sequences and a wide concentration range. As with biological samples, the more abundant ERCC transcripts have greater representation in sequencing results than less abundant transcripts. To increase representation of low abundant transcripts, we depleted 4 abundant ERCC transcripts using InDA-C. An analysis comparing the FPKM values with and without InDA-C shows a significant depletion of the targeted ERCC transcripts. This is shown by the 4 transcripts at the bottom of the graph whose FPKM values are no longer concordant. The typical slope of an FPKM comparison is 1. The increased slope of the line in this analysis indicates higher FPKM values as well as good concordance of the remaining transcripts. Analysis of individual low-abundant transcripts in the absence of InDA-C-mediated depletion, shown outlined in green, are present at an expected coverage. Depletion of the 4 high-abundant transcripts, shown outlined in red, increase the coverage of these low-abundant transcripts by 3 to 4 times. These results represent an improvement in the dynamic range of observable transcripts and is analogous to seeing greater read depth of rare transcripts in whole transcriptome or mRNA-seq studies. This method can also be used to detect rare pathogens in a biological sample, providing more informative reads and better coverage. As part of a collaboration, we generated libraries from cerebrospinal fluid in an attempt to detect the presence of the Epstein-Barr virus. In the absence of any transcript depletion, we detected 26 reads corresponding to Epstein-Barr virus. We used previous RNA-seq data to identify highly abundant transcripts. Depletion of these transcripts, along with ribosomal RNA and mitochondrial RNA, increased the number of Epstein-Barr virus transcripts detected by approximately 6-fold, providing better coverage and detection of the pathogen. NuGEN provides a number of catalog kits incorporating the InDA-C technology, with specific probes targeting human ribosomal RNA, human blood, which includes ribosomal RNA as well as hemoglobin, or ribosomal RNA from specific model organisms. Most importantly, the InDA-C probe design can be completely and easily customized for your specific project, unwanted transcript, or organism, and can be seamlessly incorporated into the existing workflow. To date, custom InDA-C probes have been designed for over 40 different organisms for a variety of studies, highlighting the versatility of the technology. The next presentation is by Dr. Maureen Peterson, one of NuGEN's product development scientists. She will discuss NuGEN's new product for single-cell and ultra-low inputs, the Ovation Solo RNA-seq system. Typical upfront depletion methods require higher inputs and are not compatible with ultra-low inputs. Incorporation of InDA-C-mediated depletion into the Ovation Solo RNA-seq system workflow allows for true whole transcriptome analysis of ultra-low inputs, plus the advantage of targeted depletion of uninformative reads. The customizability of the InDA-C probe set enables single-cell and ultra-low studies from any organism with the ability to deplete any transcript.
Thanks, Ashesh. That was a great presentation to kick off our webinar. I think you showed our audience how the use of insert-dependent adapter cleavage technology for targeted transcript deletion allows for greatly improved quality RNA-seq results. Before we move on to Maureen's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Simply type your question in the Q&A box on the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions after the final presentations. So let's get our last presentation underway. Maureen, the floor is yours.
Thank you, Ashesh, for that excellent introduction into InDA-C. I will jump in now to describing how we have incorporated it to enable a method for whole transcriptome analysis of minimal input levels down to single cells using RNA-seq. We at NuGEN recently explored whether InDA-C can be incorporated into a workflow that has been optimized for minimal input amounts. Applications for such a technology are shown here. One would be analysis of cells sorted out of bulk populations to study cellular heterogeneity. There is a large amount of interest in the fields of oncology, immunology, stem cell development, and neuroscience to understand differences between subpopulation of cells sorted out of bulk tissue. Such a technology would also enable the analysis of single-cell transcriptomics. Another application would be analysis of compromised and/or archived samples, such as RNAs that have been preserved in FFPE. These samples are typically available at minimal input levels, and in addition are often highly fragmented. A third arena for such a technology would be in the liquid biopsy field, allowing for the analysis of circulating tumor cells, cell-free RNA, and exosomes. To provide a solution for these types of studies, NuGEN has developed the Ovation Solo RNA-seq system, which incorporates InDA-C in a workflow to generate high-quality RNA-seq libraries from all sample types listed here. Shown here is a description of the Solo workflow. It is a complete end-to-end library generation solution starting from either purified RNA or intact cells through to final sequencing-ready Illumina libraries. If starting with sorted cells, we have incorporated an upstream workflow to allow direct input of cell lysates into the library generation workflow. From these cell lysates or purified RNA, DNA is removed using DNA digestion, and the entire transcriptome is amplified, followed by ligation of a bar-coded adapter. Subsequently, InDA-C is used to deplete ribosomal RNA transcripts. Additional transcripts can be targeted for specific applications at this step as well using the InDA-C technology. Final libraries can then be amplified and sequenced on any Illumina instrument. Libraries generated with the Solo workflow have been shown to be successful in discovery-based disease research, analysis of cellular heterogeneity and developmental pathways, or liquid biopsy applications. As shown here, the incorporation of InDA-C probes to ribosomal RNA transcripts within the Solo workflow is highly efficient. We tested the percentage of final library reads that map to ribosomal RNA sequences in a range of input levels from both UHR and human brain RNA controls. Without InDA-C, close to 80% of reads map to ribosomal RNA transcripts. However, InDA-C effectively reduces final library ribosomal RNA reads to less than 10%, providing superb detection of informative transcripts and providing a greater return on your investment in transcriptome sequencing experiments. We've also measured detection of genome-wide transcripts within the Solo workflow. First-strand priming enables measurement of all transcripts, not solely mRNA transcripts, enabling detection of both coding and non-coding RNA transcripts with regulatory potential. As shown here, we see consistent read distribution across various input levels of purified RNA, indicating that data gathered from Solo experiments is reliable across a range of inputs and does encompass genome-wide transcriptional information. To further measure the ability of Solo to detect transcription across the genome, we performed an analysis of all RNA classes detected in final libraries generated from two input levels, 100 picograms and 10 picograms, from human brain total RNA. While a majority did map to known mRNA sequences, a substantial portion of detected RNAs fall into the class of linked RNAs, which are beginning to be studied in cancer and other diseases and have been shown to perform regulatory functions within the cell. Various other non-coding RNA classes were detected as well, highlighting the utility of Solo in gaining a complete representation of the transcriptome and maximizing the information gained from a user's sequencing experiments. In measurement of regulatory RNAs, it is critical to preserve strand orientation. A step is integrated into the Solo workflow that allows for strand orientation preservation. And as shown here, we see over 90% strand retention from a range of input levels from two different types of human RNA. This feature allows users to assess the regulatory potential of novel RNAs identified within their experiments and look for correlations between potential regulatory RNAs and their putative coding RNA targets. Ovation Solo also provides consistent detection of genes, as shown here. In this graph, we determine the number of genes as determined by RefSeq annotation that were detected in the final Solo libraries from 2 types of RNA, again at a range of input levels. As shown here, the number of genes detected is between 9 and 10,000, even at 10 picograms of RNA input. We have additionally assessed the level of technical reproducibility from a range of input levels from purified RNA. As shown here, both 1 nanogram and 100 picogram input had a measured correlation of 0.9, and at 10 picograms it was 0.8. This high level of technical reproducibility demonstrates that libraries produced with the Solo workflow can be compared to each other across sample types with confidence that differences detected between samples are biologically accurate. As shown here, transcript coverage is uniform from the 5' to 3' end of genes, and this uniformity is consistent. Plotted here are 8 different replicates of Solo libraries generated from human cell lysates, and the distribution is very consistent and uniform across each replicate, ensuring complete coverage of genes detected. To measure the dynamic range of transcript detection, we performed an experiment using ERCC spike-in controls. ERCC spike-in controls are a set of in vitro synthesized transcripts that are present at known molar concentrations. This mix can be spiked into a background RNA template of choice and used to test efficiency of detection across their broad spectrum of starting concentrations. In this experiment, we spiked the ERCC mix 1 into 1 nanogram of HeLa RNA, then generated Solo libraries from that mixture. We plotted a measurement of detection in the resulting library of each ERCC transcript as measured by RPKM values against its known starting value, and that graph is shown here. We show an excellent correlation with an R-squared value of 0.94, indicating a linear representation of transcripts. Additionally, we measured the dynamic range of transcripts detected in the final library above an RPKM of 1 and found that transcripts from as low as 40 copies up to 9 million copies can be detected and accurately quantified using the Solo workflow. These results indicate that both highly and lowly expressing transcripts are accurately detected within the Solo workflow. Another key factor in the Ovation Solo kit is its compatibility with the direct input of cell lysates after cell sorting experiments. There is much interest in sorting cells out of bulk populations, either based on cell surface markers or simply into smaller populations, including single cells. To test the performance of Solo in such experiments, we started with a culture of HEK293 cells and sorted them using a BD-JAS instrument. We deposited these cells at varying densities into a 384 well plate with lysis buffer, and then proceeded with Solo library generation. Shown here are the results we obtained in analysis of the final libraries generated by our sorted HEK293 cells. As shown here, we tested from 500 cells down to 1 single cell as input. Across the board, we saw efficient ribosomal RNA depletion down to less than 20%. We also saw exceptional alignment rate, strand retention, and gene detection. The exact metrics, particularly in gene count, will vary based on the specific type of cell analyzed in the Solo workflow. But these results demonstrate that Ovation Solo performs well with cell lysate inputs and that it performs consistently across a wide range of sorted cells down to single cells, making it compatible with workflows analyzing cellular heterogeneity of bulk populations. We have also measured whether Solo libraries can accurately discriminate between different cell types used as input in very small populations. We considered this a test of whether true biological variability is detected using cell lysates as input into the Solo workflow. To this end, we made libraries from 5 sorted HEK293 and URCAT cells, then performed hierarchical clustering of the resulting final libraries to identify whether different cell populations were accurately distinguished. The results from that experiment are shown here. As you can see, 12 replicates of URCAT cells clustered together and away from the HEK293 library. The first order of separation is between the 2 different cell types, indicating that Solo does accurately discriminate between biologically distinct cell populations. This feature is important for accurate profiling and distinguishing of subpopulations of cells from bulk populations and highlights Solo's utility in studying cellular heterogeneity in populations of interest. We further tested the application of Solo in analysis of FFPE preserved tissue. Because many disease tissue biopsies and the relevant controls have been archived under these conditions, a workflow that can accurately capture the transcriptome of archived FFPE RNA with minimal input will be hugely beneficial to discovery research and biomarker research. We used a colon cancer quad set from Adaptor Bio, which includes RNA isolated from both colon adenocarcinoma and normal adjacent tissue. Further, each of these 2 samples is present as both fresh frozen and as FFPE preserved. Generating Solo RNA-seq libraries from each of these 4 sampled types allowed us to ask whether Solo performed equivalently with each sample type, as well as whether it can detect biologically relevant information from archived samples. Shown here are the Agilent bioanalyzer RNA traces of the input material. As you can see, the fresh frozen RNA was of high quality with RIN scores above 9 and DV200 scores above 90%. Conversely, the FFPE preserved RNA was degraded with RIN scores below 2.5 and DV200 scores of 55% to 65%. We assessed the level of technical reproducibility of Solo libraries generated from FFPE RNA samples by analyzing the correlation value of genes detected in duplicates of each of the FFPE RNA samples. Shown here, the correlation value at 1 nanogram of input is almost 0.9, which demonstrates excellent technical reproducibility using minimal FFPE RNA as input material. To measure detection of biologically relevant information within the colon cancer quad-set RNA samples, we first obtained a list of genes known to be aberrantly expressed in colon cancer relative to healthy colon tissue from a study cited at the bottom of the slide, which was published in 2008. We compared expression of these genes, as determined by their FPKM values, in tumor-derived RNA and normal tissue-derived RNA in each of the fresh frozen set and the FFPE preserved RNA set. We considered a gene to be concordant if it showed the same trend of upregulation or downregulation in both the fresh frozen comparison and the FFPE comparison. Using this analysis, we observed a high concordance rate in measured gene expression differences between fresh frozen and FFPE preserved RNA, demonstrating that Solo libraries can accurately detect disease biomarkers from FFPE archived material. We have also tested whether the Solo technology has utility as a method for RNA biomarker discovery using cell-free RNA as input material. This would enable Solo to be used in liquid biopsy type experiments where a blood or plasma sample can be used to detect and monitor disease state rather than a more invasive solid tissue biopsy. In this experiment, we used RNA extracted from human plasma as template into the Solo workflow and compared the resulting library to one generated from a UHR control. As is typical of cell-free RNA, an analysis of the input plasma RNA revealed it to be highly fragmented, with RINs of 2.1 to 2.6 and DV200 scores from 10 to 20%. Once we generated final libraries and sequenced them, we compared the distribution of RNA classes detected between the plasma RNA samples and the UHR control. As shown here, while the majority of the UHR control was mRNA, only about 15 to 20% of the plasma RNA mapped to mRNA sequences. Conversely, the majority of the plasma RNA samples fell into this other category, which includes regulatory intergenic and intronic RNAs, which can be explored as novel sources of non-coding RNA biomarkers. Additionally, piRNAs and mitochondrial sequences were much more enriched in the plasma RNA libraries compared to the UHR control. These characteristics of plasma RNA have been observed previously and are continuing to be explored. Results shown here demonstrate that Solo provides biologically relevant information from different sample types and is amenable to use in profiling cell-free RNA for various applications, including liquid biopsy experiments. As I've shown you today, there are several applications and benefits to using the Ovation Solo RNA-seq system for whole transcriptome studies with your samples of interest. The information gained is from the whole transcriptome, giving users the most abundant information in their samples of choice and opening up the potential for discovery of regulatory RNAs. Solo is compatible with generation of RNA-seq libraries from a range of input types. It can be used for biomarker discovery from archived samples, cell lysates, and liquid biopsies, giving users the most extensive array of sample types for their experiments into disease discovery and diagnostic applications. Lastly, the InDA-C technology incorporated into the workflow reduces noise from unwanted transcripts in the final library, thereby reducing sequencing costs and increasing detection and analysis of transcripts of interest. The InDA-C probes can easily be customized for specific samples, making it a versatile component of this novel workflow. With that, I would like to thank you for attending and open it up to some questions.
All right, guys, looks like we got some really great questions that have rolled in. So let's begin the Q&A session. Our first question is going to be for Ashesh. Ashesh, one of our audience members would like to know, is there a way to, instead of performing targeted depletion, perform targeted enrichment of only certain transcripts of interest?
Sure, that's a great question. The InDA-C technology is actually used for depletion of unwanted or uninformative transcripts. So while the removal of these transcripts does enrich and allow for better coverage of informative transcripts, it's not actually enriching for specific transcripts. For targeted enrichment, I would actually recommend using our targeted enrichment products, which are also completely customizable and allow you to specifically enrich for the transcripts that you may want to study.
All right, thank you, Ashesh. Maureen, the next question is for you. One of our audience members would like to know, are there any characteristic differences in final Solo libraries from high quality versus low quality RNA input?
Okay, thank you for the question. The one characteristic difference that we have observed is in the size of the final library. So in running these out on the Agilent bioanalyzer, we see from high quality RNA, our size peak is between 320 to 350 base pairs or so in the final library curve. However, with lower quality RNA, that tends to shift a bit smaller to around a peak of around 250 base pairs. That, of course, will vary based on the quality of the RNA that you put in. So moderately degraded might be slightly bigger, more degraded, slightly smaller. But in general, we do see a size shift to a smaller size distribution in the final library.
Okay, great. Thanks, Maureen. Ashish, another question for you. Someone asked, how do I generate a custom InDA-C probe set?
That's a great question. I would recommend that whenever you would like to generate a custom probe set, contact our fantastic technical support team, and they can work with you to understand your project and exactly what you'd like to do, and discuss what is needed for us to generate the custom probe set. You can contact NuGEN Tech Support by emailing techsupport@nugen.com, or you can call NuGEN directly and get to the tech support helpline.
Okay, great. Thank you, Ashish. Maureen, another question for you. Can Solo libraries be sequenced on any Illumina platform? Are there any deviations from standard Illumina sequencing parameters?
Yes, so we have successfully sequenced Solo libraries on both the MySeq and the NextSeq instruments from Illumina. They sequence well. In terms of deviations from standard parameters, we do have a custom primer, a custom read one primer that comes with the kit. That can be used if you are sequencing Solo libraries alone. However, if these Solo libraries, you want to pool them in a sequencing lane with non-Solo libraries, that can also be done. For that, we recommend pooling Solo libraries to no more than 60% of the total input. And in that case, sequencing can be done using the standard Illumina read one primer. If any users have any more specific questions about compatibility or more details about the ratios that Solo can be pooled with non-Solo libraries, again, you can contact our tech support and they'll be happy to help you.
Great. Thanks very much, Maureen. Ashish, we have a question for you. A two-part question. What is the minimum inputs required, and can I use degraded samples?
Sure. So we have several different systems. For the Solo kit that Maureen talked about, the input is anywhere from 1 cell to 500 cells, or down to 10 picograms to 10 nanograms of isolated RNA. For some of our other InDA-C kits, the input range is from 10 nanograms to 100 nanograms. And for all of these systems, you can definitely use degraded samples, and we have tested that, and it works very well. And Maureen showed you some data on how the Solo kit actually works with plasma samples, which are highly degraded.
Okay, thank you very much. Maureen, another question for you. How can an individual tell whether genomic DNA has efficiently been removed from a Solo library?
A good question, especially at the lower input range. Removing genomic DNA is really important. And there are two ways that we can tell in doing our in-house experiments. In our RNA-seq analysis pipeline, we routinely look at the percent strandedness of the final library. Solo does incorporate a step to retain strand orientation. And so your strandedness should be very high, above 90%. And that will tell you that you have removed genomic DNA. If you have not, you would expect a value for strandedness closer to 50%. And another way is to look at the percentage of your reads that are mapping to exons. That will vary a bit based on your input, but we routinely see at least 40% of our reads mapping to exons. So if that falls low, closer to the 10% or below range, we consider our libraries to be comprised of primarily genomic DNA.
Okay, thank you, Maureen. Ashish, looks like we have one last question for you. What percent of my sequencing reads will be rRNA with this system?
That's a great question. So with our InDA-C technology, we typically see over 90% depletion of the targeted transcript. The actual number of depleted transcript reads that are observed in the sequencing results will really depend on the abundance of that transcript in the starting material.
All right, thank you very much. Maureen, we have a question for you. What are the standard QC steps in validating success in generating a solo library?
A good question. So what we typically do here and what we advise our customers to do is first to run an adjuvant bioanalyzer high sensitivity DNA chip to look at the trace, the size range of your libraries. If you're seeing a nice broad peak centered around 320-ish base pairs, again, as I mentioned earlier, that can fall slightly lower if you're using degraded RNA samples. But looking for typical library distribution around that size range is important. To quantify, we advise using kappa, real-time PCR, to quantify the sequenceable library, the amount of sequenceable final library products in your product. So those are the two things we routinely do here.
Great. Thanks very much. Maureen, looks like we have another late-breaking question for Ashish. Ashish, I'm sorry. Ashish, how many human RNA targets have you targeted so far with the InDA-C system?
So that's a great question. So in addition to ribosomal RNA and mitochondrial RNA, we've done some studies where we've targeted up to 10 transcripts. And we haven't actually reached an upper limit of how many transcripts we can target. But at a certain point, depending on the cell type and what the sequencing results provide, you're not actually getting a net benefit for targeting additional transcripts.
All right, thank you very much, Ashish. And it looks like with that, we have come to the end of our webinar. I'd like to remind everyone that the webinar will be archived on our site for 6 months at www.genengnews.com. So if you missed parts of it, you can watch it again, or you can feel free to forward it to your friends and colleagues, which we always recommend. I'd like to thank Maureen and Ashish again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks for NuGEN for sponsoring this webinar. So hopefully we'll see you again soon at another Gen webinar in the near future. Goodbye for now.